Fantastic. Okay, well, thanks for everyone coming. We've got what should be an interesting talk today. We've just been chatting to Mark, deciding which talk he wants to give. So we're really flying by the seat of our pants, which is excellent. That's what we want from our speakers. Mark's a long-time collaborator at the Institute. He's done a lot of work with the Barley Group, particularly Robbie, and he's published well with Robbie. But we tried to convince him to come away from the dark side and speak about something else today. So let's see if that happens. Mark's well known in the area. He's an interesting background, and he's now at the King Abdul University of Science and Technology University, where he holds two hats. So he's a professor of plant science there, but he's also the co-founder of a company called Red Sea Farms, and he'll allude to that. And I think we're going to get him back to talk about the commercial piece on that as well, because I think that's equally relevant, particularly for our flagship projects. One thing I'm going to throw at Mark before we even start, and I was looking at his interview for Trends in Plant Science, which he's probably forgotten all about, which was many years ago. He said he was asked what's the best advice he's ever been given, and it was focus. Let's see if it applies today.
Do as I say, not what I do.
Absolutely. Right, Mark, over to you, and we look forward to hearing you speak.
That's a great introduction, Derek. Thank you. Yes. Oh dear. So I'll share my
screen. We can kick off.
Yeah, that's great. Thank you. I have a terrible habit of not focusing, which is why I laugh like this. But I am interested in plants. I'm a plant guy, and thanks for that introduction, Derek. And I'm interested in how plants work. For example, why a plant down by the sea is a completely different species to one that's even just 100 meters inland, what genes are in that plant that helps it tolerate those environments compared to the ones perhaps in the more mesic fields. And then, of course, seeing if we can apply that knowledge to agricultural systems. Originally, a lot of my focus was about trying to improve food production. Increasingly, I've been concerned more about the sustainability of food production and also the quality of food production. I don't mean quality for rich Westerners. I'm talking about the quality of nutritional value for, in particular, people in developing countries, which is the world's biggest health problem. But anyway, I'll try to stick in this seminar, try to focus on salinity tolerance, which is my primary area of expertise or interest. The background for this, at least for me over the last decade, when I moved to the Middle East, is that it's really quite a stunning thing I've learned in this last decade, is that a very substantial fraction of our food is produced under irrigation in irrigated systems. The estimates are 1/3. You hear people talking about over 40%. It's a lot by whatever the number really is. And also incredibly interestingly and alarmingly, about 2/3 of all the water we use as humans is used for agriculture, contributing to putting that food on our plate. There's been an amazing set of work done in NASA over the last 15 years, where they put up a pair of satellites called the GRACE satellites, which were looking at the differential gravitational pull on a pair of small satellites that were launched and orbit just a very short distance apart from each other, a couple of kilometers apart from each other. And the difference, the millimeter differences in height due to the gravitational pull can be measured. And from that can be deduced, and it's done repeatedly, spinning around the world, taking measurements, and ground truthed, of course, and calibrated to a certain extent. This is documenting actually all sorts of interesting things, like how much the Greenland ice cap is melting, but also relevant for this talk, how much the groundwater is being abstracted in major aquifers around the planet. And it turns out that every aquifer, every major aquifer that is being used for agriculture is being depleted. Given that a large fraction of the water that is used in irrigated systems is coming from groundwater nowadays, surface waters have been fully exploited pretty much. This is really quite alarming. In other words, we're mining water for producing a large fraction of our food. So a large fraction of our food is on the water budget unsustainable. Things have to change. And that's the big context. Actually, just Pakistan, a country where I have quite a strong interest and work. There's the area of land. Oh, this is an odd, I've changed this slide in my other presentation, Derek. 6 million hectares of salt-affected land in Pakistan, where the yields are being significantly affected by salinity. That's equivalent to the total arable land area in Scotland, which, as I understand, is about 10% of the area of Scotland. So just to give you a context, that's it's quite a large amount of land being affected. And in Pakistan, it's incredibly intensive systems with a very, very large and rapidly growing population. Anyway, it's serious stuff. Oh, and this is out at the front of our field station, one of our maiden field sites close to our university. It's a farm across the road from the field station, and it's had to be fully abandoned. This tree's now fallen over. This is completely rusted out, and it's happening all over the place. The groundwater's going down, the salinity goes up, and the system breaks down. There's heaps of water on the planet, of course, but 99% of it that is usable by humans is salty, or potentially, sorry, usable, is salty or inaccessible. And only 1% of the water is fresh that we can access. Although, of course, 2%, twice as much is frozen than is fresh. So I guess global climate change will help with that. Bring back Donald Trump. So anyway, what we have to do, or what a small contribution we're trying to make towards this really quite serious global imperative, is to see if we can improve our ability to access water that we currently do not access, in particular salt water, brackish water, or even ultimately seawater. So if we're going to unlock something, we need a key, and the keys, from my perspective, and again in the context of this talk, are technologies. And there's two major technologies which I think we can really use, very, very powerful, and actually extremely well wielded at the James Hutton Institute, I should say. And one of these is what I call the new genetics, which is the genetics turbocharged by genomics. And the other is huge opportunities with phenotyping. And I think together, as phenotyping becomes much higher throughput, much smarter, catching up, if you like, coming along behind the amazing revolution with genomics. But as it comes along, I think this does provide us with huge opportunities for serious innovation. And one of the things that we've been doing for the last decade or so is seeing what we can do to try to unlock the fabulous genetic diversity that's in wild germplasm. And we can do this by trying to unlock wild germplasm and use this to try to make existing crops more salt tolerant, which I have to confess is the approach that I've been taking for most of my career, and not exclusive. Another option is to get plants that are very, very salt tolerant and then domesticate them, turn them into crops, an approach which we're doing increasingly at the moment. So one example of work which we did a long time ago now when I was in Australia, in Adelaide, was collaboration with CSIRO. It's a fantastic collaboration with Rana Munns. It's like the grandmother of the field of salinity. She came to me actually when I was still in my lab in Cambridge with some bags of durum wheat, which had very low sodium accumulation. Durum's quite a high sodium accumulating wheat. We've now understood why that is at a molecular genetic level. And it turned out that the couple of bags of wheat that she brought with low sodium accumulation in the shoot had come from an old cross with a wild diploid relative from the Middle East, done by a PhD student back in the late 1960s, and had been sitting in a drawer ever since. That student was trying to bring in disease resistance, novel disease resistance alleles. They had accidentally brought in a gene which we identified called HKT15 on B chromosome, which was helping the plant retain sodium in the roots. And CSIRO, Rana Munns, Richard James back-crossed this using marker-assisted selection while we were working out the molecular side of it, the physiological side of it. and then started doing field trials with, say near isogenic lines is slightly overstating it, but lines which are pretty cleaned up with just pretty much just the gene, the causative gene introgressed. There are actually two genes, but one gene was the main one. The other one came with too much drag and actually caused yield penalties. But with one of the genes, NAX2, the sodium excluded number 2 gene, had, when grown in low salinity parts of the field, had no yield penalty, but when grown in the high salinity parts of the field, actually had a very significant increase in yield. So the plants were having an increase in salinity tolerance due to this increased ability to maintain low shoot sodium in the face of high salinity. Beautiful aspect of this work, dumb luck, if we're being honest, was that there was no yield penalty in the low sodium parts of the field. I then I've been writing an annual review, another annual review of plant biology the last couple of weeks. And it's all about trying to actually deliver salinity tolerance into crops. So I've been talking to the breeders about their progress with this material. Australians look like they're not that keen on it. And I think this is partly because the primary drivers for wheat breeders in Australia are not for the more marginal fields. Most of the yield, when you consider it nationally, most of the yield is coming from the higher yielding fields, the ones that are less salt affected. This is sort of just mathematically obvious. And so breeders really do focus on increasing yields in the high yield fields, which is understandable when looking at either the profit of a company or even the national interest. However, national interest in terms of export dollars. However, lots of farmers work these marginal lands, and lots of farmers would benefit significantly at the farm level and have actually proportionally quite significant increases in their profit if we were able to roll this out. So this is an interesting little, I'm not going to go into public versus private, but different motivations for perhaps a university researcher versus a company or even a national interest. So I think in Australia, these genes haven't really been grabbed enthusiastically, shall we say, by the privatized breeding companies. Even the Durham program in Australia now is privatized. But interestingly, Richard James, who did a lot of the breeding work in Australia and the physiology actually at CSIRO, he's collaborating with Bangladeshi scientists, and that's being introgressed there, and looks like that actually is going to be delivered into the farmers' fields in southern Bangladesh to provide a wheat intercrop between the rice. So you do the rice in the wet season, you can do a salt-tolerant wheat in the dry season. That's delivering that. Another example, which hasn't been delivered, is here, where we're looking at other traits. So there was a review we wrote a few years ago, in 2008 actually in annual reviews again, which was on physiology of salinity tolerance. That was Rana and I did this review. And we sort of looked at 3 primary traits that were contributing, we hypothesized were contributing primarily to salinity tolerance. And when I came to KAUST, I started to think a bit more about how you could dissect the plant, the life of the plant, and look at the effects in different ways, and look at the effect of salinity on some of those other things. And inspiring me was John Passiora's analysis of drought tolerance, and how you could look at the yield of the plant being the outcome of the product of the total plant growth, the water use efficiency of the plant harvest index. And so I looked, we started having a series of research projects, a series of students looking at these 3 components. And it's in moderate success. We're working with barley and rice. And this is an example I have just from the rice work, because we've been following up at the molecular level from this, so going in and characterizing the causative gene. So here we've got beautiful little signal in a rice association mapping population. It was Ausch rice. It was mapped many years ago by Susan Cooch, and we actually were collaborating prior to publication with her, and published at a similar time some use this mapping population. And we found here that there was an association on chromosome 11 in rice and the trait was not just transpiration use efficiency. We did find loci for those, but this was the one which was interesting me, which was not just transpiration use efficiency, but it was actually an interaction where we're looking at the ability of the plants to maintain transpiration use efficiency in saline conditions relative to non-saline conditions. And it turns out that we seem to be zooming in quite nicely on a little set of 5 tandemly repeated genes in that locus. They're a protein kinase of unknown function, and where, you know, what it comes down to at this stage, grinding away doing the molecular characterization of these genes. Okay, those are 2 examples of where we've been trying to look at traits that contribute to salinity tolerance and seeing if we can bring them into crops and improve the salinity tolerance of these plants. But I was starting, especially in this tough environment. When you're working in North Africa, Middle East, it's West Asia. It's a very, very tough environment. And when you start doing the types of work that we've been doing, I mean, it's fine, and I'm proud of the science, but I do wonder about really how much impact we're really going to make in these types of environments. And you know, started to think about other crops and whether we should, perhaps it might be, you know, making more step changes by getting wild plants that are very salt tolerant and domesticating them. So the first little attempt at this I did was actually like a halfway house, was with quinoa. And with quinoa, we've got a plant which can grow on half seawater. It's drought tolerant. It's a remarkable plant, native to the Andes, where there's a lot of very saline soils. And it's already been partially domesticated. And we wanted to both look at it both in terms of as a model salt tolerant plant that we can study the mechanisms of tolerance of, but also as a plant for which we might be able to help contribute to further domestication. So quinoa is an amazing plant. It grows in areas where nothing else grows. So these are a few photos I took on the shores of the world's largest salt lake, which is also, I think, the world's largest source of lithium, which I would hope might, probably won't, but might lead to an improvement in the lot of the Bolivians. But out there in the real Bolivia, it's very, very low rainfall, very poor irrigation infrastructure, and also very poor abilities to operate and maintain irrigation infrastructure should it be installed. So you'll see fields like this where there's a very scanty scattering of quinoa, and the fields are very hard to mechanize because they're full of rocks and very uneven, and a lot of it's the kids that should be going to school who are working in these fields. But it also is a crop which has huge yield potential as well. When we're doing field trials, these 3 photographs in the bottom right-hand corner are taken from field trials we have up on the Tibetan Plateau and in southwestern China, and it's remarkable how much you really can get out of these plants when you give them some good conditions. So anyway, we wanted to start to try to do some genetics in the field for quinoa and start to look in particular at traits which seem to be not completely a focus of some quinoa breeding programs, and we hope we'd be able to provide some resources to underpin some of this. So we did a genome. We've now resequenced actually over 1,000 lines, and starting to do some association genetics in field trials, and trying to look for traits which will help with mechanization, yield stability, and of course yield in its own right. The plant's got terrible early vigor. It's only really doesn't have that much dwarfing. It's often got quite large branching, very plastic as well. Herbicides, weeds in the fields, just a huge problem. It'd make a very big agronomic contribution just by getting herbicide resistance into these plants. In the context of the Middle East, It's a plant which originates from 4,000 meters in the Andes. Is really where a lot of the center of diversity of this plant is, and it's very sensitive to heat. And it's got lots of other great abiotic stresses, but its real Achilles heel is its heat sensitivity. And so this is our primary focus for our work now. Now we've done a lot of this foundational work. And we've got a wild relative. It's actually a tetraploid wild relative, probably the immediate progenitor of domesticated quinoa. And it grows wild in the lowlands of Chile and in northwestern Argentina, in the foothills of the Andes, where it's hot as hell. And we've got a collection now, I think, of 100 accessions of Chenopodium hirst, and we've resequenced these. And we're now got a project to quantify and introgress heat tolerance into domesticated quinoa. Okay, so genomics has been done in particular, well, it was originally done by David Jarvis, who now has his own faculty position in BYU in the US, and his work was carried on by Elodie Ray, young French woman in my laboratory. So she's done a fantastic job and has really tidied up lots of the genome. We're actually about to submit a paper manuscript. I'm just looking out the window at her going, where is that manuscript? Should be on my desk any day now. And that's looking at the as a second version of the genome. I think it's a very good one now. So the two subgenomes, it's an allotetraploid. It's an ancient allotetraploid, and probably occurred 1 million years ago or more. And there's very good now syntenic alignment between the two subgenomes, and between we also sequence the putative diploid progenitors, or at least the closest relatives we think of the diploid progenitors. There's good alignment. There's some very clear couple of rearrangements, in particular in the B subgenome, but I won't go into those details. The resequencing, you end up with some really out of my depth here, some population genetics where I'm not sure I completely understand some of the analyses, but there may be a reason for this. But at any rate, you can get between 6 or 8, depending on how you analyze it, putative populations. But it's quite interesting, because what we can do with this type of analysis is you can identify within each of the likely subpopulations, and we're doing this now, is lines which have got minimum or no admixture. So we're getting like the pure, if you like, representatives from each of the, I think actually we've chosen 8 subpopulations, and then are doing a de novo gold standard genome sequencing for those. We actually got within these, we actually combined it also with the phenotyping. So we got the pure members of each of these subgenomes, and then looked at the field phenotyping that we've been doing over the last 5 years or so, and getting plants which were looking reasonable and not crazy wild or crazy tall or something like that. So trying to get some reasonable, phenotypically reasonable looking plants that are also pure representatives for each of these sub groups. And that's work that is hopefully going to be published quite soon. These types of arrangements show that some of the population structures, in a way that I can understand more. Here we've got a very clear falling out into 2 subgroups, which are identified for a long time as being quite distinct. Lowland quinoa, primarily from Chile, and then an highland quinoa, primarily from Peru and Bolivia. And what's interesting is that a lot of the wild sequences fall out between these 2, which tempts one to think that there may be some 2 independent domestication events. But there's also a lot of after domestication reintrogression from the wild. So I think you have to be pretty careful. So actually got a nice project where we're looking at some ancient DNA from cave sites, some old grains, and also stalks, and seeing if we can try to piece things together a bit more. Okay, this is just more representation. It's interesting, the 2 highland and lowlands seem to have a different type of linkage disequilibrium. But it is very tempting to speculate that we've got two. Quinoa actually was domesticated twice, perhaps once in Chile and once maybe northwestern Argentina or southern Bolivia. We really don't know yet. It's all history there. But at any rate, it's very, very interesting, provides useful raw material for start doing some genetics. And so we've been doing some phenotyping in a range of different locations around the world. We're doing this now for many years, and I think it's starting to get interesting. So we've been phenotyping in quite a lot of locations with quite a lot of collaborators. I'm just highlighting here David Wu in China, up on the Tibetan Plateau, Qinghai Tibet Plateau, and here in actually an Australian collaborator, and never met him when I was in Australia, a guy from Western Australia called Mark Warmington. So we've now got, so that's the site in northwestern Western Australia, and these are actually two sites in China where we've really been getting a lot of phenotypic data. But we've got collaborators all over the world now who are using this mapping population. Okay, if we're going to have these mapping populations, so we're trying to define this genetically, but we also need to define the phenotyping of these plants as well. So we've been trying to develop an international standard. We actually end up publishing this in like an international consensus paper. I think it was published just a few months ago. And this is where we've got these phenotyping cards developed actually by one of our technicians, and just common sense. So trying to define and have it usable in the field. Oh, here's the paper here, with a lot of authors, with a lot of a whole phenotyping consortium we put together. I think it was about 50 people involved. So trying to get things so they're more uniform and sort of acceptable internationally. And that's fine for sort of, if you like, normal phenotyping, but of course we've got wonderful technologies now, and so we can do things with drones, and with computing power the way it is now, you can do some pretty incredible things where from drone images you're able to do 3D reconstructions like this, and you can do this even with multispectral cameras and start to try to extract information from the fields, and that's proving to be quite interesting, especially when you combine it with machine learning. So I've got a fantastic collaborator here in Caus called Matt McCabe, who specializes in this type of work, and his research scientist Casper and PhD student Jocelyn have been doing their stuff over our fields, and then of course you then try to do the genetics on top. This really is a bit like just a status update at the moment, because these analyses are still ongoing. And Clara, PhD student here working on the quinoa, is actually right in the thick of these analyses at the moment. But you can try to get predictor variables from multiple mass analyses. Now, some of this starts to get quite tricky if you have big issues around data management, as well as, of course, the computing power that you need for some of these huge data sets. And this was one student, I admit I've just slipped slyly sideways just for one slide, so forgive my bad language, to Arabidopsis. But I had students working with 5 different species, sort of doing similar stuff, and they were realizing that they were all having the same problems with data management, and got together to develop software. So here at Mariam, it was one student with one relatively small project doing an association mapping of salinity, the effect of salinity on photosynthetic traits, and she ended up with populations, lots of traits, lots of plants, because you can automate all of these measurements. And so their data nightmare, they got together and fixed themselves, and led by one of my former postdocs, who's now faculty at Cornell, Magda Jukovska, and they developed this thing called MVApp. So for any students in the audience, I really commend this to you as a tool to help you. It's one thing to gather data, it's another thing to be able to manage it and analyze it, and we're really trying to help. And this was student-led. Yeah, okay. So you do your phenotyping, do your genotyping, and then you do, of course, your genetic, your statistical genetic analyses. I get very nervous doing this in front of audiences such as we have here, where you've got such fantastic work. But at any rate, we're doing it in our relatively amateur way, and starting to find some interesting things. We think here we've got a gene controlling height, and I've highlighted this because we have plants that is quite photoperiod sensitive. It's a facultative short day plant, quinoa, and so when you're growing them at different day lengths, you can get quite differences in height, and so the genetic analyses also come out different. Sometimes it's stable, like this locus on chromosome B7. It's absolutely the same marker from two completely different studies, one in northern Germany growing through the summer, and one in northern Australia growing through the winter. And then another locus which is completely vaporized, disappeared. We can sort of validate that we're on the right track generally by going straight to genes very quickly for some traits for which there's some biochemical and molecular information such as saponins and flower color. And here we've got ones controlling, talking about the days to flowering and height being linked together. Don't think that's such a big surprise though. Okay, so we're starting to do some work with quinoa and starting to try to do a bit of genetics in field sites around the world. It's taken quite a few years to get the foundation there, but as I say, I think we're there now, which is good. Now, quinoa is good, and it's very salt tolerant. It's got some good stress tolerances. But I wanted more. I'm sitting here in the Middle East where you've got no permanent surface water, no significant recharge of deep aquifers which are being extracted, and the only sustainable source of water ultimately is the sea. So I started to think about whether we really could use seawater for even just modest agricultural systems. And so I started to work with Salicornia, which is an incredible plant that can grow actually better in full strength seawater than it can with no sodium chloride. And the plant is able to accumulate very high concentrations of salinity. There's heaps of genetic diversity in the plant. We have started to try to do some genome sequencing. In fact, Vanessa, a new postdoc with me, has really got a grip on this, so that we've got good now genome assemblies for at least 2 species of Salicornia. And okay, I'm wanting to do applied work and wanting to deliver and try to make an impact, but I'm still a scientist with a lot of curiosity as well. And when we look at Salicornia, it blows my mind how it's accumulating huge concentrations of salinity, maybe up to 1 molar concentration of sodium in the above ground parts. And like rice plants will start turning brown with like 30 millimolar or something. So it's able to have accumulation of 30, 50 times the concentration of sodium in its shoots than a rice plant. That's amazing. So I'd love to know, and that has to involve subcellular sequestration of sodium. So I'd love to know the transporters which are responsible for this in the vacuolar membranes of the shoots of Salicornia. So I had a PhD student do this, and took both transcriptomic, see what's being inducible, and a proteomic approach, where we've got a continuous sucrose gradient, and took 12 fractions down a continuous sucrose gradient from a membrane enriched fraction isolated from these shoots, and did that with plants that were not salt stressed and with salt stressed plants. I'm looking for salt stress induction of transporters or repression of transporters. I can explain why later if you want. Pretty surprising results immediately just by looking at markers and trying to validate this with Western blotting as well, where as you go down the sucrose gradient, classically you'd have the tonoplast up in the lighter fractions, plasma membrane at the middling fractions, and then the thylakoids and mitochondria, the subcellular organelles, down to the heavier fractions. And interestingly, the plasma membrane and tonoplast were swapped in their locations down the sucrose gradient. And we've been very careful with this. This is definitely the case. And they stay there even with the salinity, even in the absence of salt stress. So, one can speculate about why that may be, and perhaps because this plant's actually got a lot of proteins in its tonoplast membrane to keep that sodium inside the vacuole. We honestly don't know, of course, but that was just that was really a nasty, well, it was a nasty surprise at the beginning for me and the student. But when you start analyzing the salt induction of these tonoplast allocated, plasma membrane allocated proteins, really, and he did this lovely PCA analysis where you can see the tonoplast enriched tonoplast proteins really coming out separated very, very nicely, with the plasma membrane and endoplasmic reticulum on one side and the organelles on the other side of this sucrose gradient. So we got really quite nice, and looking with high confidence at the salt-induced proteins, he identified 151 proteins in the subcornea tonoplast. So he and I went through these in great detail and identified 63 proteins that he tried to clone to express in yeast to see if they would affect yeast. He managed to successfully clone 46 of these genes encoding these proteins, likely salt-induced vacuolar proteins, and ended up with some results which are still in progress. Again, we're still in a progress report stage at the moment, where some proteins were definitely increasing the salinity tolerance of yeast, including things like a sodium proton antiporter, or predicted sodium proton antiporter. This is actually a ribosomal stabilizing protein, which is pretty interesting as well. An unstructured and disordered protein. Okay, so that work actually, if anybody's going to referee it, we're putting in a grant application, right? Like literally on Sunday, it's the deadline for putting in a grant application to build on this work and really try to nail exactly which genes are, which proteins, I should say, are in that vacuole and helping that subcornea lock away its proteins, lock away its sodium. Sorry. Okay, so that's just a quick jog through some of the plant science. Now, working here in the Middle East on salinity is great because we can make a contribution to reducing the pressure on the consumption of fresh water in this very, very dry part of the planet, and of course, hopefully be able to move this technology elsewhere. In the meantime, there was a PhD student. He started in the center here, the Center for Desert Agriculture, and he was an agricultural engineer, and he was working on cooling greenhouses and trying to stop using fresh water for cooling greenhouses. So here, when you've got a greenhouse, you it's pretty crazy, but what you need to do, well, what people have been doing, sorry, is spending money desalinizing water and then like an hour later evaporating that water to cool a greenhouse. And it's a huge use of energy, very large use of water, and it just seems completely crazy. And so he did his PhD developing systems for being able to cool greenhouses using seawater. Now, in some ways, it's almost trivial. It's engineering and there's some tricky stuff in there, but it's not rocket science in many ways. But it turned out to be more complicated than one might expect because the seawater is down in the sea. And funnily enough, and down by the sea, it's a lot more humid than it is inland. And so that elevated humidity, and that's particularly so in this environment where it's hot, and that elevated humidity has caused a lot of problems and greatly reduced the efficiency of the evaporative cooling, and especially at night when the relative humidity can go up to well over 99%. So you have zero cooling at night time. So he did his PhD on that. And I was actually on his committee, his PhD committee, and at the end of his PhD, he did a brilliant job and had a fantastic defense. And I went to him afterwards and said, why don't we get together and make a company? You're trying to substitute freshwater use with saltwater, so am I. Let's see if we could get it together. And so that was 4 years ago, and that started Red Sea Farms. So Red Sea Farms is a company which is dedicated to trying to reduce the environmental footprint of agriculture, and it was founded. I just explained the foundation. The foundation was to try to reduce the fresh water footprint by substituting as much fresh water as possible in the system with seawater or other salty water. We've since broadened very rapidly into other sustainable technologies, in other words, technologies that are reducing the environmental footprint. We've been working in particular in controlled environment agriculture. Now, I have to admit that this was because of Ryan's PhD, which was with greenhouse cooling. It turns out I think that this is a very, very good choice for more reasons than we realized at the time. And I think when you're looking here in Saudi Arabia and the long-term sustainability of agriculture in this type of very hostile environment, you really only got two options. One is to grow stuff in seawater, and that really, let's be honest, is a modest option. And the other, just because of the area of land that you could use, that you can still maintain and manage and not allow salt to build up in. But then the other option is to be using covered controlled environment agriculture. It really is the only two options in the long term. And so it was a good option for local relevance, but it's also very important, I think, globally. And I think controlled environment agriculture is one area and one option that's extremely powerful for sustainable intensification, where we're trying to grow more with no more land, with fewer inputs, reducing CO2 emissions, and increasing efficiencies. And so this is really, really important. There's another dimension to this as well, which is to do with the nutritional value of the foods. I don't know how many of you are familiar with the Eat Lancet Commission that made a report on healthy diets and sustainable food systems. It's published a couple of years ago. I actually happened to cotton on to it just when it was published, and I went, wow, this is amazing. It's turned out that it really is an amazing analysis that's really getting a lot of attention. I wish it got a lot more attention actually globally. But basically, this is the global average. We eat 3 times as much meat as is good for our own health. This is health focused. We're eating 3 times as much starchy foods as is good for our health. Plus, we're wasting about 1/3 of our food, wasting and losing about 1/3 of our food. And we're under eating by about 50% fresh fruits and vegetables, which is actually one of the most important sources of many of the micronutrients, whose paucity is the world's biggest health problem. I mean, forget COVID and HIV or whatever other headline grabbing. There's the biggest health problem by a mile, the biggest number of single deaths caused by, and disabilities is caused by micronutrient deficiencies, insufficient iron, vitamin A, iodine in our diets. And the best way to address this is by increasing our intake of fruits and vegetables in particular, and having less red meat production. So these are the predictions of what we need on the planet if we move towards a healthy diet. In other words, less of the red meats and starch and more of the other stuff that we actually need, providing the micronutrients that so many of us are lacking, and halving the waste, not eliminating the waste, but at least halving the waste, which is technically not unreasonable. When you do this analysis, we actually don't need to increase cereals production. We're already growing enough cereals for 2050. But we're growing nowhere near enough fruits and vegetables. So for me, this analysis is actually very important, very persuasive. It's just almost parenthetical to this particular part of the talk, but it's very interesting. We also need to grow a lot more legumes, they're saying. Okay, any rate, so I think covered environmental systems are important. for multiple reasons, both from sustainable intensification, reducing in this region water footprint of our food production, and for health. If we're able to, that's the other point I want to make. When we're wanting to increase fresh fruit and vegetable production, fresh fruits and vegetables have short shelf life. They're high value, short shelf life. So if you're sitting here in a hot, dry environment, really the only way to get your fresh fruit and vegetables is to fly them in. It's a massive carbon footprint. I mean, we calculate it in our company. It's extraordinary how high the carbon footprint is for flying fruits and vegetables around the planet. I was really shocked when we were doing the numbers, and so if we can grow these locally by developing the technologies to enable them to be grown locally, that's going to improve human health, and it's going to reduce the carbon footprint of the supply of fresh fruits and vegetables to the population. So yeah, very happy. And I can't say I was smart and thought of all this before we started the company, but I think it is an interesting and important analysis. And so we just set this up. We're starting with growing grafted salt-tolerant tomatoes in a greenhouse cooled with seawater. We clean the roof with no water, and we end up with a system where it's in the order of 90% of the fresh water that would normally be used for the production of these fresh fruits and vegetables is actually substituted with seawater. We need water in the system, but we actually are able now to replace 90% of the fresh water with salt water. And that's pretty good. So in this environment, when you're growing tomatoes in an open field or in a conventional greenhouse, you need about 300 to 350 liters of water to produce a kilogram of tomatoes. That's like 4 big tomatoes. And with our systems, we're still getting the numbers, of course, and we're still getting better and so on, but it's looking like it might be in the order of 20 liters for a kilogram of tomatoes rather than 300. And that's pretty good. So that's the sort of thing that we're doing. And we're bringing together engineering and plant science and IT to develop these saltwater greenhouses. And how are we for time? Oh, that's probably enough. So yeah, there's the freshwater footprint going down. And that's the other thing. You look on the internet and you see, vertical farms and all this sort of stuff. People say it's fantastic, and they'll emphasize how it doesn't take up much land or it doesn't take up much water, whilst failing to say how much energy that it consumes. And you can get the same in the greenhouse where, we can save huge amounts of water in our greenhouse by using mechanical cooling, failing to say that that's actually using huge amounts more of energy. So here you can save water by using mechanical cooling, but it comes with a massive energy cost. Whereas if you simply, in inverted commas, substitute fresh water for salt water, you can break that food water energy nexus that we're currently trapped in. I think that's pretty good. And it comes with a business case that's solid because, final point, because we're being pragmatic. We're not sitting here as sort of pious or idealistic academics saying we're going to have no fresh water use in our greenhouse. We're going to be only using renewable energy. You can sit there and make those statements. Technologically, you can do it, but it doesn't make business sense. And so nobody will buy it. It will not propagate. You won't have to take out patents to protect it because people don't want to steal it because it's not a business. So what we're doing is we're saying just making a small pragmatic step backwards and saying maybe 50% solar and maybe 90% salt water, still using some fresh water. That way it actually becomes, we can make a business case that's viable. And that's what we're out and doing now. So we're trying to develop now. Yes, we started with saltwater cooling, but we've now got a whole series of technologies that we're actually piling up more of these to try to address the environmental challenges that we're finding in the region. Okay, so thank you very much for listening. This is a huge number of collaborators around the world. It's fantastic fun doing this sort of work, and I've got a fantastic lab. This is how we were 2 years ago, happily having our Christmas party on the beach here in Saudi. Of course, that's how we have been the last 2 years. I'd just like to thank all my group. I've been trying to acknowledge people with their names in the corners of the slides as we've gone through. And thank you all for listening. Thank you.
Thanks, Mark. That was excellent.
Okay.
I'll refer you back to that comment I said. Did you focus? No.
I've tried to have some thread, but, yes.
Okay, so we've got questions popping up. It's always good to let other people ask them. Carmen, are you still there? Do you want to open your mic and ask your question? Is Carmen still around?
Hello, Mark.
Hello, Carmen.
Great talk. I wonder, well, if what are the traits that the more prominent traits that make quinoa drought tolerance, and if you have spot other pseudo cereals or pseudo cereals that are also good for domestication.
Yep, excellent.
Okay.
Thanks, Carmen. So to answer your first question, quinoa actually it's got very good sodium exclusion. It's a plant which has, it seems, very efficient transporters to help it maintain a low shoot sodium, like wheat. So that seems to be the primary mechanism. It has on its leaves very prominent bladders, but these bladders have got nothing to do with salinity. Unfortunately, there's quite a body of literature out there which says these salt bladders, and then they go straight into transcriptomics and all this sort of fancy molecular stuff, forgetting to actually check whether they accumulated salt in the first place, and they don't. So these bladders on the leaf are probably to do with either defense against insects or other predators, and also it may well be involved in UV protection as well. Remember, these things are up at 4,000 meters, and you get the bladders around the apical meristem and the young developing leaves and the flower buds. So I think they've got other uses, but they've got nothing to do with salinity tolerance. In terms of domesticating cereals, I have a fabulous colleague here. He's only been here a relatively short time, Robbie Waugh. Robbie Waugh. Jesse Poland. Sorry, I put them in a similar category. Jesse Poland. Jesse Poland's a wheat breeder from Kansas State, and really a young guy, adopts technologies quickly, rapidly, aggressively. He's really good. He's come here, bought into this local problems very, very interestingly, and is already starting to work on the distichylous genus of grasses. So these can grow in seawater, and they have salt glands, two-celled hairs on their leaves, which are like microglands. They've also got very good sodium exclusion ability, remarkable plants. And there's one species, Distichylus palmeri, which has actually already got quite interesting, reasonable grains. And so Jesse's exploding into that field very quickly. Yeah.
Okay, good.
So I like what he's doing.
I'm sure Robbie will take that compliment that you've confused him with someone considerably younger.
Yes, well, that's true.
Raul, do you want to ask a question?
Yes, impressive presentation. Thank you very much. So I see that there is still a lot of progress in understanding the tolerance and tolerant and escape mechanism to salinity. I mean, that's the plant side. So my question was more related with the technology to the salt water. So how close or how far is this technology to the other work that we are doing with the plants? Because one of the things that I learned when I was working with salinity is what are the level of water? I mean, the salinity and the water that you're using for experiment, but later you explain that in your Red Sea farm.
Now, I'm not sure I'm completely with you in your question. So you're talking about if you've got like a saline soil that's dry versus a saline soil that's got a higher water
the level
content.
of water that you are you using in your experiment are similar to the level of salinity that you get from the desalinizations?
So you talked about diluting the salt water for your Red Sea stuff. How much are you diluting, or diluting out the molarity of salt.
Okay, yeah, I gave absolutely no details on that. I'm sorry, on the plant side, because I was focusing on the engineering. I focus on the engineering because, and I had another talk actually prepared for maybe we do it in a year from now on Red Sea Farms, especially focus on the engineering, because to be honest, the engineers are the ones who are making the huge contributions to water saving, because it's the engineers who use all the water in the first place. In the greenhouse system, about 90% of the water that's used is for cooling in this environment. I mean, obviously not in European environments, but here it's so, you know, I can sit here and go, it's 90% salt water. And I'm very careful to say that's for the system. And then I do focus on the engineering, not the plants, because it's the engineers who are doing all of that water saving, or they're doing by far the majority of the water saving, the engineers. We do grow the plants on brackish water, but it's only about 1/10 seawater. It's not that salty, all right? We're trying to improve it. Well, we are improving it. Just got to make it commercial. And that's because we've had some fantastic breakthroughs, I have to say, with tomato rootstocks. So we've been developing rootstocks that are conferring fabulous levels of salinity tolerance on the scions. And that work now is in field trials and greenhouse trials. And I'm particularly excited about it when I go to northern Egypt, where there's saline fields and farmers who are trying to do everything right, who just don't have the genetics, and they need the genetics. And so we're getting rootstocks, and part of the problem, I can tell you, is just getting it so it is economically viable for those farmers, because you have to have the seed produced cheaply. This is all F1 hybrid rootstock stuff. So when you look at a Maxifort seed, it sells for 30 cents. It's crazy. That's because of its high cost of production, partly because of its high cost of production. So we're working out how to generate that F1 hybrid seed more cheaply, so then we can deploy it in the farmers' fields. So we're at that sort of level now. But yeah, we can get plants growing on 1/5 seawater tomatoes economically, actually probably even more than 1/5 seawater, because of the rootstocks.
Thank you.
I haven't published any of that yet.
So if I can ask a question about the Salicornia work, I mean, that
Yeah.
hyperaccumulation is mind-blowing. Is that something you can actually use as a natural cleanup system for brackish water, or an intercropping, or a...
No, I mean, you can do the sums, you can do the math fairly, relatively easy. The problem is it's a small volume of biomass.
Oh, okay.
And so the actual total amount being removed is relatively small. We'd need to have a poplar tree or something like that accumulating those sorts of volumes. Yeah.
So you're thinking more then of just pulling out mechanisms and genes and the transfer across to more important plants?
Yeah, we do have one candidate gene, and we're dropping it into rice and tomato. One of my students is very excited about this. I'm sure it's needed more than one gene, unfortunately, but let's see how they go. I mean, let's see how we go. But it's that is the dream, and I guess hopefully one day, in 20 or 50 years from now, the world will actually need GM and GE. It will have to have these tools of biotechnology.
It's changing at the moment. I mean, and also looking at that, on these growing brackish systems or very salty systems,
yeah,
I presume you're measuring salt uptake in the plant and the bits we may eat. Are they Is the salt just increasing? Because I mean, the Western diet's not got a great interaction with salt.
No, that's a very, very good question, and it's actually the plants. Plants like humans will do anything to protect their children, and so their children are their seeds inside the fruit, and so the plants will invoke every mechanism to keep the salt out of the fruits.
That's good.
What we find with tomatoes, it's quite well established, is that if you give salt to the tomato plant, the fruits actually accumulate a lot more sugar. So they're actually sweeter, and they're actually more acidic, because they're having to increase the acidity to pump the sugar into the vacuoles and the berries. And so the tomato fruits. So they will do everything, keep the salt out, which then gives a water balance problem, Raoul. And so to counteract that water balance, they actually accumulate more soluble sucrose, fructose, and keep out the salt, and not accumulate as much starch inside the fruit. So the fruits actually taste awesomely good when you're growing them on salt water.
So I'll maybe just ask a last question, and it's picking up a bit that you stated. I think maybe people might have missed that. When you were talking about the delivery of solutions in Australia to
Yes.
tolerant ones, but they just didn't get taken up. So creating the solution is not everything.
No.
Is there a reason why they didn't take it up? Was it just because they just didn't want to farm on that type of land?
Yeah, well, that I sort of tried to make the point. I was going a bit fast, so I was also ad-libbing, because it's in my head at the moment, because it's worrying me, because I don't want my life's work just to be a bunch of papers. Oh, actually, there's a bunch of wonderful students as well, of course, who come out of my lab, which is probably the best legacy an academic can have. But I'd quite like to have something a bit tangible on the farmers' fields as well, if possible. And so I've been worrying about this, and yeah, I think in a system which is wealthy like Australia, which is very high production, very high export, to be honest, and actually I did the sums, it's something like 98% of the grain produced in Australia is produced from fields that are 2 tons per hectare or more. I know that number's laughably low in the European context, but in Australia that means a reasonably high quality field, and so the breeders are breeding for that, whereas the salt affected fields are down at 2 tons or below per hectare. And so those are actually large areas of land, and they're farmed by a lot of farmers, but they're poor. And I'm not saying nobody cares about poor people, which is probably half true, unfortunately. But when you're looking at the national interest in terms of the export dollars, or when you're a private breeding company who gets your revenue by the amount, in Australia they have an endpoint royalty system. So, there's no point in supporting those farmers because you don't make much revenue from that. Of course, for the farmers and us as people who are thinking more about the system rather than just the raw profits that a company has to think about, it's sort of almost our duty personally to deliver these. And so in my review, actually, I'm going to exhort myself, our colleagues, to actually take these things through ourselves. And some academics do that, and then they make an impact. I mean, there are academics who do take their stuff and they believe in it and they take it through, and then they're helping a minority of people who are otherwise ignored for perfectly logical reasons, I should say, by the breeding companies or even by national governments.
It must be an age thing, Mark, because I think they're both born in '63, and my mindset's kind of similar now. It's creating legacy, and actually, papers are fine, but I want to see something out there.
I publish papers because I'm supporting my students or my postdocs. That's for me the only reason now.
I want to see practicality, and if you're following that route like you're doing, where I'd say you've got skin in the game, and you do tend to work that bit harder for it as well.
Yes, I really strongly support people doing entrepreneurial activity, and I think it's what you said, spot on. You put skin in the game, and you end up actually really. I tell you, it's a damn good ride. It's a very, very interesting exercise that I've gone through.
Okay, so on that note, if everyone can show their appreciation in the normal route, and I think what we'll do is we'll definitely have you back to talk about Red Sea and just the translation of science into company, because that's a different talk, but equally valid to a lot of the younger career ones that are keen to see that as an alternative route.
Absolutely. Good.
Hopefully
Thank you very much, Derek.
Hopefully we can meet soon. So thanks to everyone. And the talk will be uploaded on the APGC website at some point shortly, I think. So again, thanks, Mark, and hopefully we can speak soon.
Thank you very much, Derek. Thanks all.